I think we'll get started. So good afternoon, everyone, and welcome to the fourth and final session of our Early Career Investigator webinar series. I'm Janice, Director of Communications for the Stem Cell Network. On behalf of the network, I do want to thank everyone for joining us for this important webinar series. It's the first we've done for early career investigators. We've hoped that you found it useful and informative. And I also do want to give a special shout out to our sponsor, ThéCell, for supporting this series. This week is going to look a little different as we've both a keynote speaker and a panel discussion. So we've got a lot to cover. I'll get started right away by introducing our moderator for today. Dr. Nika Shakiba is an assistant professor at the University of British Columbia. Nika completed her postdoctoral training in Drs. Ron Weiss and Domitila Del Vecchio's labs in the Department of Biological Engineering at MIT, and her doctoral training in Dr. Peter Zantz's stem cell bioengineering lab when he was at U of T. Now at UBC, Nika's group applies systems and synthetic biology approaches to reverse and forward engineer the competitive interactions between stem cells in order to answer fundamental biological questions and give rise to a novel class of engineered stem cell therapies for RegMed applications. Nika, I'll hand it over to you. Thank you.
Thanks so much, Janice, for that introduction. So as Janice mentioned, this is our fourth and final session of the webinar series, designed to cover some of the main challenges and issues that investigators tend to face in the early stages of their career. Wanted to give a quick thanks to Stem Cell Network for developing and hosting the series, and a special thanks to ThéCell for sponsoring the event. This is meant to be an interactive event. We really want to hear from you, so please use the Q&A feature to submit your questions for our panelists. So as Janice mentioned, we're going to have an extended session, and we're very fortunate to have a keynote speaker. So to kick that off, I'd like to invite Dr. Julie Fradette, who is the director of ThéCell Network, to introduce our speaker. Dr. Fradette is a researcher at Quebec Laval University since 2005 and a full professor of the Department of Surgery at Laval University School of Medicine. Thanks so much for joining us.
Yes, well, good afternoon. I'm really pleased to be here, and ThéCell is pleased to join the Stem Cell Network in hosting this seminar series. And I attended all of the sessions, and I really want to applaud the leadership of Stem Cell Network in putting together this very interesting program. So as Nika mentioned, we are starting off a little differently because we are very fortunate to have Dr. Jeff Karp with us today. Dr. Karp is a professor of medicine at Brigham and Women's Hospital, Harvard Medical School. He is also a principal faculty member at the Harvard Stem Cell Institute and an affiliate faculty member at the Broad Institute at the Harvard MIT Division of Health Sciences and Technology. If you look a little bit into Jeff's work, you'll see that he's working in so many fields: the drug delivery, medical devices, stem cell therapeutics, tissue adhesives. He has over 100 issued or pending national and international patents. Several technologies developed in his lab have led to multiple products currently in development or on the market, or for the launch of 8 companies that have raised over 400 million in funding. So technologies include high-tech skincare, tissue adhesives, and 3D printed biomedical devices, immunomodulation with biologically responsive materials, small molecule regenerative therapeutics, cannabinoid therapeutics, biomedical devices to improve child safety, needles that automatically stop at their target, and a bioengineered luminal coating for controlling GI targeting. So I'm sure you will have some selected examples, but from that list, if you have special interest, you can go and look more into his work. So Jeff has received numerous awards and honors. The Boston Business Journal recognized him as a champion in healthcare innovations, and MIT's Technology Review magazines also recognize Karp as being one of the top innovators in the world. 3 members from his laboratory have subsequently received this award. So his work has been selected by Popular Mechanics as one of the top 20 new biotech breakthroughs that will change medicine. So we are really thrilled to have you with us. And please, Jeff, the floor is yours.
Wonderful. Well, thank you so much for inviting me to speak today and for the nice introduction. Before I get going, my institution requires that I disclose my conflicts of interest, for which I have many. As you heard, I've started a number of companies, and I am a consultant for all of them, and I also hold equity in these companies. And today, what I thought I would do is share a little bit about the strategy of my laboratory. I have, and I consider this, really a highly iterative process, and something that, I've just really tried to develop an awareness of and take an active role in kind of changing things and experimenting with how I run my laboratory. Every single aspect of the lab I've been experimenting with over several years, and I've come to a few realizations that I wanted to share with people. And so one of the first is that it is really, we hear a lot about, it's all about the people and hiring the right people to come to the laboratory. And I feel like it's much easier said than done. And well, we have a goal of really hiring people who are just, super ambitious and really excited, about the field of biomedical engineering and the potential it can have, and people who show a lot of potential, and also, get along well with others. I feel it's difficult for me to judge character, because almost everyone I'm presented with, I feel like, I want to hire them. And so what I've done in my laboratory is I've set up a hiring committee, and that committee, interviews candidates, and actually I've set it up so they can veto me. They really have the say of who can come to the lab and who should not. And I think that's really important, because the people in the lab are really, they're not going to want someone to come who's going to be competitive or who has too much overlap with their expertise. They're going to look for people who can fit into the lab, but also can bring something new to help elevate their science. And, I read an article some years ago that said that when you interview people, you really try to hire yourself. And after I read that, I started to notice that was actually the case. And I think there's a lot to be said about diversity, which I'll talk about in a moment. So I feel, having a strategy of hiring has been really important. And in fact, there's a couple of people in my laboratory who have just been outstanding at it. So I make sure that they're involved in every kind of new recruit that we bring to the lab. Another thing that I found is really important is really trying to find ways to empower the people in the laboratory. And what I mean by this is really try to find ways where people in the lab can truly spread their wings. They can take control of their projects and really run with them. And just to give you an example, and there are several, I had a PhD student in my lab, Ben Mead, who joined a few years back, actually recently graduated, and he's now a postdoc at MIT. And he was very interested in writing grants in his first year. So we wrote some grants together, and then the grants got funded. Actually, 3 grants got funded. And he said, well, what do we do next? And I said, well, you need to hire a team. So in his second year of his PhD, he had 3 full-time people working in the lab. He was taking courses, but they were reporting directly to him. And so that allowed him to develop his mentorship skills and really, push the envelope of his professional development very quickly. And in addition to that, in terms of empowerment, one of the foundations invited me to speak at to their board about an update on our project. And I knew if I asked them if I could bring Ben with me, they would probably say no. So I just brought him into the room, and then I presented the first few slides, and I handed it over to him, and he presented the majority of the work. And later on at the reception, the people on the board were just really impressed with Ben, and again, it was one of these opportunities where he just really was able to unleash his potential, and I think that, to me, that's really what empowerment is all about, is trying to find ways for the people in the lab to really just take charge, to push the limits of their skills, and have them, iterate their own strategies, as early as possible. So that I found has been really important. Another thing is I have a lot of affiliations, and I look at my affiliations really as access to resources. And so, I have affiliation at the Harvard Stem Cell Institute and at the Broad Institute and at MIT through the Health Sciences and Technology program. And this provides us with really, almost seamless interaction with people at all of these institutions, as well as access to core facilities. And so, to me, it's really about, we really want to minimize any rate limiting steps to getting things done or accessing new tools or resources. And in addition to this, we're constantly always trying to write grant proposals to bring in new funding to the lab. So we'll always be able to start up new projects and advance in the directions that we want to advance. In addition to that, I've populated my lab with people from really highly multidisciplinary backgrounds. And so we've had engineers, biologists, we've had a gastrointestinal surgeon, a cardiac surgeon, a dentist, multiple MDs in training. And I feel like the diversity is really important for a number of reasons, one of which is we're constantly brainstorming because we're running into all kinds of challenges in every project. And what I envision is people sitting around a table and everybody can provide a unique perspective. If people have different, experiences or expertise, they're going to have different ways that they look at things, and they're going to have different skill sets and different tools that they can bring to the table, different, knowledge of different areas. And so, I feel that is really important because when someone, chimes in and has ideas, usually no one else has those ideas. And then that validates that person, they're more likely to go all in. And I feel it's very similar. We've had people from 30 different countries in the lab, and that multi-cultural background, I find, the diversity of people from different countries has been really important. Because people think differently, there's different education systems, and so they're going to approach problems very differently. And then also what that does is there's, each country kind of has their own sort of medical issues that they face. And so this allows us to then learn about all of these other medical conditions and not just the ones, in our backyard. And then another thing that I do is I like to commit to being a mentor for life for anyone who joins my laboratory. And I think that this really is important because it gives people that comfort level to really go all in when they come into the lab. And so I tend to write letters for people when they leave the lab, as well as review sometimes grant proposals or all sorts of different things. And I just feel like that's really important to the whole kind of circle of life of the research world. And then just to talk a little bit about the translational strategy for the lab. Again, this is something I've been iterating, and it's definitely a work in progress. But I really want to, I really try to start each project in my laboratory with the goal that at some point we'll be able to move something to clinical trials and eventually to patients. And one of the things that I've realized is that advancing on a basic science project versus basing on a translational project is very different. We definitely need the basic science to be in place. So that really has to come first. And then once we have the basic science in place, we can start to think about how we might define the problem further to advance a translational approach. And often when we start projects, I like to kind of go in with almost like detective hats on, where we assume that the problem that has been defined has not been defined the right way by others. Now, it might have been, but if you go in with that sort of mentality, then what happens is that you tend to, turn over every assumption. You tend not to take anything for granted. And I think that's really important when starting projects, is to assume that, all the assumptions are wrong. And you really want to make sure that these assumptions, you want to have confidence in all of the assumptions that have been made. And then you're not, subject to the dogmas that exist within the field. And so that's often where we start the project, is really trying to define the problem and then conduct experiments that will confirm some critical things that other people have found, and then ideally try to find something new, try to gain a critical insight into the problem. And one of the things I tend to ask in every single lab meeting is, what's the bar that we need to exceed to get people excited, to get potentially our colleagues excited and investors excited, the whole field excited? So it's a pretty difficult question to answer, but I think it's one of the most important to ask from a translational perspective. Because it gives you a target of where you're headed. Now, often we'll see, in basic science projects, we'll see, there's bars, and we're showing like, there's statistically significant differences, and the differences look very large. But those results often may not be important from a translational perspective unless we're comparing to the gold standard, let's say. You're not always going to find a gold standard, but if there is one out there, you want to include it in your experiment. So, for example, we've been doing some work in immunotherapy, and so we're looking at the specific models that others are using, and we're asking the question, okay, what percent of animals, what's the best result that anyone has ever achieved in the different models, and then how much better do we need to do? So maybe that's people have achieved before us 20% survival for 6 months. So, we need to figure out whether our goal is going to be, 30% or 40% for 6 months or a year. Like we like to define that early on. And then what that does is it just aligns everything in the project towards that goal, to try to really make a beeline. Now, we may not achieve that goal, but I feel when you set that as the goal, you start to uncover some critical things that are important to achieve the goal. And sometimes we find that we don't know enough biology to get to that goal. And then we need to focus on elucidating that biology. Another thing I found has been really important, perhaps maybe one of the most strategic decisions I ever made in my career, is kind of transitioning out of my postdoc. I realized that, I had a lot of just incredible mentors who had started companies and were moving things out of the lab, but I feel it's another thing to do it yourself. And so kind of lacking the translational expertise and the entrepreneurial skill set, what I did was that I made a decision to meet with people in the translational ecosystem. And so literally every 2 or 3 weeks, I've been meeting people, be it entrepreneurs, reimbursement, regulatory experts, manufacturing experts, people in companies at all sorts of different positions. And I've been with the goal of not just meeting them, but forming relationships with a subset of people. And then these people that I form relationships with end up almost serving as an informal advisory board for my lab. And so as we're advancing on projects, I'm constantly interacting with them and asking them, translational questions like, do you think this could be manufactured? Or, if we were going to set up a clinical trial, even though it's way into the future, what would be the comparator? What would the clinical trial look like? Do you think investors would be interested in this? Like, what kind of result do you think we need to achieve to really, get people excited? And so I'm constantly engaging the community to ask these types of questions. And then another thing is I like to try to, solicit this feedback, not just in the beginning of projects, but along the entire route. And I think it's just so important because there's so many, holes in our thinking. And I think it's really important, something actually that I've realized recently. I realized it for a while, but I couldn't really figure out how to say it. But what I figured out, I think recently, how to say it, which is when we define problems, I feel we have a tendency to define the problem as a biological problem or as a, let's say, a medical problem. But if we really want to translate, we have to consider the patent problem, the regulatory problem, the reimbursement problem. There's there's all sorts of things that we need to think about, manufacturing. And so I feel we need to kind of cast this wide net over what the problem truly is, and it needs to include a lot of the downstream things that happen, after let's say one starts a company, and what are all the things that need to occur? Because I think there's been so many things that have been promising, but haven't made it to humans because it's just too complicated. It can't be manufactured, or the clinical trial would just take too long and too many patients, and no one would pay for it. Or, there's all of these considerations, or it's hard to get a patent on, this particular new technology. So I think, it's just really important to get this feedback along the way for all aspects of the problem. And then we really try hard to publish in the best journal we can. And I think that's important to try to generate press. And, it's not essential, but I think it does help a lot. And so I've made an effort to get to know a number of journalists that exist at a number of the media outlets. And I think that's important because there's been a number of times where we publish a paper and then, companies reach out to us and ask, is this available for licensing, or is there collaborative opportunity? And I think that's really important to try to create that type of gravity when you're moving towards a translational effort, just because it requires so many resources and so many different types of expertise. And then also trying to find an entrepreneur to lead it. And actually what I found is by developing relationships with entrepreneurs, over time, committing to it kind of as a process, it's much better, because when things mature, then you have people you can reach out to say, are you available? Do you know someone who's available? Whereas if you just wait until kind of towards the end of a project when you have something up and running, working with people, especially in a company, it's like, kind of getting married to them in a way, and you can't form those tight relationships very quickly. They have to be formed over, longer periods of time. And so I think it's a commitment to developing relationships very early. And then as things progress, then you can rely on some of those relationships to find people in the community who might want to, join the effort and move things forward. And then, we're always kind of, we have minimal rules, I'd say, overall in the lab, and we try not to have too many rules, but sometimes, we need to create them. And one of the rules that we've created is that when we start new projects, we don't like to take anything existing that we have, because often the problem definition is completely different. And when you force something that you already have, often it doesn't work very well. And you find out kind of later than you'd like. But there has been some projects where we have found technologies in the lab when a new problem presents itself, and that technology is almost a perfect fit. So there's all these, so I feel like it's like anytime we've tried to kind of create rules on advancing things in the lab, we find certain conditions where those rules need to be broken. So I wanted to share with you just a few stories from the lab where we started from problems and then tried to develop new technology with a clear problem definitions in mind. So going back over 10 years ago, Dr. Pedro Del Nido, who's chief of cardiac surgery at Boston Children's Hospital, presented us with a challenge that he was facing in his treatment of patients, and that's septal defects. So a certain percentage of children are born with congenital heart defects, and then a percentage of them require surgery. And so these are holes in between the chambers of the heart. And so he said to us, sometimes I go to suture this tissue and it's so fragile, it just tears. And he said that, there's devices that work really well in adults, but you can't downsize them because the materials are non-degradable, and the heart just will outgrow the implant. So he says, is there any way we could work together to try to solve this? So we came up with this idea of having a patch that we would put inside the beating heart. It would attach to the tissue around the hole, immediately attach and block the hole. And then cells would migrate on top of it, form new tissue. The material would degrade, and then the patient's left with their own tissue sealing that hole, which could then naturally grow with the patient. And so, this seemed, reasonable, but at the same time, extraordinarily challenging because there were a few key, issues that we needed to overcome. One was this is an incredibly wet environment filled with blood, has all kinds of cells and enzymes. And so every surface in the heart is coated with a layer of blood. So how were we going to get this to attach to the tissue and not just the blood? Another is that there's a lot of expansion and contraction cycles going on here. So the material would need to be somewhat, have to be elastic. And then you also have the constant shear of the blood that's rushing against the surface, which potentially, could delaminate. So there are all these challenges that we were up against. And so we kept running into one wall after the other, and at one point just almost stopped making progress. And I feel like this happens in every project, and now we almost expect it to happen. And what I've noticed, just trying to reflect on this, is that when we approach problems and we don't get the result we're looking for, and then we step back and approach the problem again, often we apply the same thinking, but we expect different outcomes. And just to really highlight this point, I want to show you the following problem that occurred in Europe. This actually really happened, where here's this car that's been fallen into the water, and probably a few different options, but the problem solvers lined up here decide to bring in a crane, and things seem to be going well until the following happens. So one problem has become two problems, and the second problem is even bigger than the first one. So the question is, what are the problem solvers going to do next? And you can see them here. Looks like they're trying to avoid getting splashed. So what are they going to do next? And the answer is they're going to do the same thing that all of us would do next, which is apply the same thinking that we originally did to solve the first problem, which is bring in a bigger crane. And what do you think happens? The same thing. So the question here is, how do we break free from the conventional repetitive process in our minds? I feel like, our minds tend to gravitate towards these low energy processes. So it's almost like, when you are developing a habit or learning a new skill, your brain goes into this kind of high energy state to learn the skill. But as soon as you've learned it, then your brain goes into this low energy state where it just likes to anticipate what comes next. It kind of goes into this robotic mode. And I think there's many examples like this in life, where essentially we can almost like think of something else while we're doing something, completely different. And I think that this actually works against us when we're trying to solve problems, because it limits our ability to activate our minds and really look beyond what's available to us or what we've learned, to look for new ideas and solutions. And I think there's many ways to break free from this repetitive process. One way we've done that in the lab is to turn to nature for inspiration. And it's really this idea that everything that's living, every plant, every animal, if it's alive today, it's here because it has solved an insurmountable number of challenges. The evolutionary process is a problem-solving process, and arguably the best problem-solving process around. And so what we've done is as we define problems or challenges that we face, we've started to ask the question, well, can we find examples in nature where this where this problem may have been, or similar type problem has been solved? And if we can then learn about those solutions, and there's many described in the literature, we might be able to come up with ideas that we would never have otherwise thought of. And so kind of going back to the problem at hand, we started to think, okay, well, what creatures exist within wet dynamic environments? And we kind of zoomed in on a couple. One is sandcastle worms exist within the sea, and they sit on rocks and the waves hit them and they don't move. And then slugs and snails, sometimes you'll see a snail sitting on a leaf and it's raining, and the rain's hitting it, but it's not moving. Or, sometimes you'll see it walking along the ground and there's this kind of goo behind it. So we realized that these creatures that can exist within wet dynamic environments have a couple things in common. One is viscous secretions. and anything that's viscous has inherent adhesive interactions with its substrate. And so, if I put honey, for example, on a table and then try to wash it away, it takes some time to rinse away. And we thought that would be useful because sometimes when you're placing a, let's say a medical patch into place, the surgeon's not always going to get it in the right location immediately, and they might need to kind of move it around. So if we could have perhaps a viscous precursor, like some sort of pre-polymer glue, then the surgeon might be able to put it down into a location, move it around, and it will stay in place until we sort of cure it to its final state. And we've actually developed a lot of light curable materials, and we started to think light could potentially be interesting here. The other thing that was interesting about these viscous secretions is that they contain hydrophobic agents, and things that are hydrophobic repel water. And so we were thinking, well, that might be interesting. And it kind of goes against what others were thinking in the field at the time, which is if you're going to make a tissue adhesive, you want it to be hydrophilic. You want it to have properties that are like the tissues in the body that have a high water content. But this was telling us, well, wait a second, what if we make this hydrophobic or has hydrophobic component? Could we then repel the blood away from the surface of the heart so that we could have intimate contact with our glue and the tissue? And so we decided to advance with that, but there was another challenge, which is how do we make this really, stick strongly over the long term? And for this, we turned to ivy, anyone who's tried to pull ivy off a building or wherever it is will know that it actually requires an incredible amount of force. And the mechanism was elucidated about 10 years ago, where ivy has these root hairs that go up and down a surface looking for crevices, and then they insert into the crevice, and then they secrete a glue, and then they dry, they mechanically kind of shrivel up and interlock. And so that gave us an idea of, well, what if we could, there were ways that we could kind of chemically attach to a surface, but we weren't sure whether that was really going to work because it would just be fouled in the presence of blood. So, chemistry approaches often will break down under biological conditions, such as the ones we were looking at. So we thought, well, what if we could get this glue as soon as it contacted tissue to infiltrate into the tissue? And then we know light can penetrate tens of microns, maybe 50 or 100 microns into tissue. So we might be able to then apply light afterwards and cure the material so it hardens, and that might just work. So we went through many iterations. There's no recipes in the literature for how to get material to infiltrate into tissue. But with that being the goal in mind, we eventually figured out how to do it. And here's an electron micrograph showing when we applied this to heart tissue, we put the glue or the adhesive onto the tissue, we cure it immediately with the light, freeze fracture it, and here you can see the adhesive on top and the collagen fibers in the tissue, and you can see that the glue is nicely kind of interdigitated in between the collagen fibers to give us this almost like a tissue-like Velcro, so it's mechanically interlocked with tissue. So I want to fast forward to one of the most challenging experiments we've ever performed in the lab, which is an animal experiment. So if anyone's kind of squeamish at the sight of blood, you may want to look away. So here we made a very large defect in a rat heart, and this is with a dermal biopsy punch. We have a suture here that we're going to remove. It just the animal doesn't bleed out during the procedure. And then we also had to develop another material, a patch, which was biodegradable, elastic, and transparent, so light would go through it. You'll see here we have the patch. We don't really wipe away too much of the blood. We apply the patch. There's a thin layer of glue on the underside. We get it into place, and then we shine the light for 5 seconds. Now, something went horribly wrong with this particular experiment because we made the patch too small. It was one of the first animals we operated on, and it kind of slipped a bit. So while it attached, there was this massive leak. So we're freaking out trying to figure out, what to do. And someone said, well, why don't we just scrounge up as much glue as we can find? So we put that onto a spatula, and then we applied it directly to the leak without wiping away any blood. And if you look carefully, you'll see that because the glue is hydrophobic, it's actually repelling the blood. It's less red right here. And because it's viscous, it stays in place long enough for another pulse of light. And then we end up with a perfect seal. And we took these animals out 6 months and they all did fine. So we then decided to advance to an even more challenging model, which is, would this work in a large animal like a pig? And could we attach this inside a beating heart? And so for this, we used a device that Dr. Del Nido had developed. We put our patch on the end, placed this, made a small incision in the myocardium, placed this up against the septum, shined the light, and then removed it, sutured the myocardium. And here we have 2 pigs on the left and right. Here's the patch here and the patch here, 82 beats per minute. And if the patch wasn't attached, you wouldn't be able to see it because it would be moving around. And then we came back at 4 hours because we wanted to ask, well, what if the heart rate goes up? Would the patch still remain attached? And at the 4-hour time frame here, you can see the patch on the left and on the right still attached, 165 beats per minute. So really excited that the patch was able to remain attached even under these dynamic conditions. And then we came back after 24 hours, and here you can see the patch. There's a suture here, which is part of the deployment mechanism. But to us, while this was kind of a early days for this technology, it was really a big leap forward because we were able to show that we could get this patch to attach inside a beating heart, many beats of the heart over the 24-hour period, and this even withstood this increased heart rate. Now, we've since, with colleagues at the Wyss Institute and MIT, Connor Walsh and Ellen Roach, developed an approach to deliver this patch via an interventional procedure. So we don't have to make an incision in the heart to deploy it. We can actually place this into a blood vessel and then target the heart with it, put the catheter through the hole and deploy the patch. But at this time, I want to just pause for a moment and acknowledge the 2 co-leads on this project, Nora and Maria. Nora is a cardiac surgeon from Germany, and Maria and Nora was in Dr. Del Nido's lab, and Maria was in my lab, and she's a material scientist from Portugal. And they just had this incredible synergy, this diversity of expertise. And I think really the reason why we were able to advance this project as far as we could, they just, were really able to feed off each other's energy and provide this complementary skill sets. And now Nora, we started a company based on this technology. Nora is a cardiac surgeon in Germany and an active consultant for the company, and Maria is the chief innovation officer at the company that we co-founded together. So the company's name it's called Tisium. It's based in Paris, and they were able to take the glue component of this and apply it for vascular reconstruction. We decided to keep the septal defect model in the academic setting, but because the glue was so promising for a number of applications, especially for sealing blood vessels, we were able to get regulatory approval in Europe for vascular reconstruction. And the company now has set up a GMP manufacturing facility to bring this to market. And in addition to the glue, the company has also developed a number of devices to deploy this. So you can spray it onto tissue here. We've added a blue dye so you can see it. In the middle, you can see this being applied underwater. So this is in a water tank with a vacuum assisted device. And then here, via minimally invasive type settings, we can apply this through a small bore catheter. And then on the left, you can see the light that we're applying. This is LED light, so it's very safe, minimal temperature change, and it can be applied in a pen kind of like structure. So it's very easy to apply this light on the surface or potentially anywhere in the body. So I was giving a talk on this technology at the New England Aquarium. Actually, it was a really cool conference called Zubiquity, where it brought together animal and human doctors. And this was the penguin exhibit. And it's probably one of the most painful talks I've ever given, because as I'm speaking here, there's the speakers and there's penguins here. I mean, they might be a little hard to see, a bit washed out, but they end up getting very rowdy because of, the speakers and we're kind of invading their home. And so I was afraid that no one would hear what I was saying. Actually, I had to stop like 6 or 7 times during my talk But luckily, Dr. Bill Rosenblad, who was in the audience, heard. At the time, he was chief of dental and craniofacial medicine at Angel Memorial Hospital, which is really a leading veterinary clinic in Boston. And he said, I think this could potentially be useful for a number of applications. And so a few months later, he contacted me and said, he has a patient, it's a 10-year-old bulldog, and it has a hole from the oral cavity to the nasal cavity because he had pulled a tooth and it left a big hole. And he had done 3 separate surgeries to seal this hole, but they failed each time within a few days because there's all these forces in a bulldog's mouth. And what he had done is he had cut some tissue, pulled it over the hole and sutured it. But it just tore within a few days for 3 separate procedures. And so he said, well, what if we applied your glue and cured it in place and then pulled the flap over top of it, the tissue flap, then the material would provide some structural support. So it would take some of the forces off of the tissue flap. And then maybe that would degrade over time and the tissue flap would provide cells and new tissue would form. So we wanted to go ahead with this. And here's what the hole looked like. It was being infected and the animal wasn't really eating much and really had hit the extent of what was possible in veterinary medicine. So I mentioned this to Dr. Yuhan Lee, who's an instructor of medicine in my lab. And next thing I know, I take a look, I walk into the lab the next morning and there's this cow head sitting on the lab bench. And I'm like, Yuhan, what's going on? And he said, well, here, come check this out. And so we had never tested whether the glue would work in the oral cavity. And so he applied it to the mucosa. And so here you can see he's kind of applying a spatula to remove it, and it's actually attached very well. So this was kind of our N of 1 before we went ahead with this procedure with the animal. And so what we did is Dr. Rosenblad debrided the hole. He applied several layers of the glue. Here's the tissue flap. You can see it's been used before. He cut underneath it and then pulled it over top. And then here you can see the sutures that are in place. And so, so the animal went away and came back 3 weeks later. And remember, 3 procedures had been done that had failed on day 2 or 3 or 4. And so Dr. Rosenblad's like wrestling with the animal, trying to open its mouth, finally opens it. And I'll never forget this moment because he was almost crying, like he was getting very emotional because it appeared to have worked. Now, this was only 3 weeks, but you can imagine this had been his patient for, probably 10 years. And to have so many failed attempts, kind of get up each morning to do this procedure, and then have it fail and potentially work was really an incredible moment. So this actually ended up working for quite some time, and the data that we were able to collect, I think, is really, really pivotal. So here is the animal before the procedure, and you can see incredibly unhappy. And then here he is after the procedure. So, gone to a very, very happy. So gone from here to here. And this dog's name was Poppy because the owner liked the slugger from the Boston Red Sox. And so I, like to Google everything. And so I was looking around for Poppy and I found this image and I was like, that's why he called him Poppy. And then the local news got wind of this and showed up and wanted to film Poppy on this operating table. So this was actually a really neat experience for us because we never really had thought that we'd be able to help pets. Always thought humans and it would take a significant amount of time. This is just an N of 1, but I was able to go to Angel Memorial and do grand rounds, which means I basically gave a talk there very early in the morning and have kept in touch with a number of clinicians. And now we're actually talking about another project in the lab, potentially applying to pets early on to see if we can go beyond what is possible in veterinary medicine with some of the experimental approaches that we're working on in the laboratory. So I think this is also something that people can keep in mind for their own work, and it might be useful to get in touch with some of the local veterinary clinics nearby where you are to see if there's opportunities to help. And then finally, I just wanted to talk a little bit about a project that we've been working on for quite some time. Lots of twists and turns. We actually started up this project in the intestine and ended up in the inner ear because of just the massive societal problem with hearing loss. Hundreds of millions of people have severe hearing loss, and we really don't have many options other than cochlear implants, which are incredibly invasive and suboptimal, and hearing aids, which really, can be beneficial to some patients, but overall, there's a lot of improvement that's needed. And so we started thinking, well, maybe there's an opportunity to have a regenerative approach for hearing loss, and we dug into this further and realized that it actually was the hair cells as one of the key cell types that are critical for hearing that are compromised in people who have hearing loss. And so all of us are born with about 15,000 or so hair cells per inner ear. That's in the shell-shaped structure called the cochlea. And these are not hairs, but rather these stereocilia, these little projections, and they move in response to sound waves. And then the cell converts that to an electrical signal that goes to a neuron and then to the brain. And what happens in hearing loss is actually these cells get damaged and then they die and they go away and they're never ever regenerated. So hair cells, the 15,000 or so that you're born with in each ear, that's all you got. And these cells can die via exposure to noise, as well as certain types of drugs, certain types of antibiotics or chemotherapeutics can kill these cells. And we knew from kind of looking around in the literature that birds and amphibians like frogs and toads possess the biology to regenerate these hair cells throughout life, which you can see here. But in humans, these cells never ever regenerate after birth. And we knew that humans contained progenitor cells that remain in place even after the hair cells die. But those progenitor cells, which can form new hair cells, actually never divide. And so we were interested to ask the question if we could develop an approach to activate the progenitor cells that exist in situ with small molecules. So could we apply small molecules potentially to the middle ear, have them diffuse across the round window membrane, which is the entrance to the cochlea, to then activate the progenitor cells to divide and then form new hair cells. And so we identified a couple molecules in the intestine that could regulate intestinal stem cells. And intestinal stem cells have a marker on their surface called LGR5. And so through regulating the Wnt and Notch pathways, we were able to significantly amplify the numbers of intestinal stem cells. We then tried that on the progenitor cells in the inner ear, which never divide. And we were able to demonstrate the same molecules could significantly amplify numbers of these progenitor cells, and they were able to form lots and lots of hair cells. In fact, we were able to form something like 60 or 70-fold more hair cells from a single mouse than had previously been demonstrated. And so we started a company called Frequency Therapeutics, which was able to demonstrate the first ever hearing regeneration with this approach. And they were able to show that this work has been published, so you can look up the paper here. So through doing extensive studies in a rodent model, and then there's also some clinical data that was included in this paper, we were able to show that indeed we could regenerate the hair cells and functionally restore certain levels of hearing. And so the company was formed in 2015. They did a first clinical study in Australia, a very small study there, in 2017, and then they initiated a clinical study in 2019, a phase 1/2 trial, which is being conducted right now in the United States. And patients who had hearing loss for many months, the Frequency was able to show that they could improve hearing in several patients. And their hearing was substantially improved. Here you can see some of the data from one of the trials. And this is data where initially the patients who have had stable hearing loss for months and months, they come in, they have 50 words read to them, and then they repeat the word And if they get the word right, then they counts as getting it right. So here you can see with the blue bars are the baseline. And so, some came in with 14 or 7 or 20 words to begin with, but then after a single injection of these 2 molecules into the middle ear, after 90 days, they were able to recognize many more words. Frequency was able to show that several of the patients who had reported improved hearing, that hearing had persisted for up to 21 months after that single injection. And now the company is in the process of exploring different dosing regimens to try to figure out what's the best regimen to maximize the effect. So this is still early, still lots to learn, but I think there's going to be a lot of promise here in this arena where the idea is to take small molecules that can target progenitor cells or stem cells and manipulate them in situ. So I think this is probably a much simpler approach than expanding cells ex vivo and then transplanting those cells. While that's very promising, I think there's huge opportunities here to really, through understanding signaling in the niche, to then replace those signals with small molecules as ways to bring about a new form of cell therapy without the cells. So at this time, I would like to really just highlight 2 major tools that I've described. One is Bioinspiration, and we're not like copying nature, it's not biomimicry. Rather, we're focusing on a problem that we've encountered, and then we're turning to nature for some new ideas. And then we're going to improve on those ideas to really advance the problem at hand. And then something else that I've realized that I think is just really a critical tool is radical simplicity. And it's this, just notice that often in academia, there's a tendency to incorporate more complexity into things. And sometimes you can get into better journals if you have more complexity and you're able to achieve something. But I think that when you're trying to translate technologies to patients, there really is something to be said about maximizing simplicity wherever possible. There's been countless technologies that just haven't made it to patients because they've been too complicated. You just can't manufacture, you can't do the quality control. And so we're constantly looking for ways to simplify our approaches. So I just want to thank everyone for your attention, and at this time I'd be happy to answer any questions that you may have. I see a couple coming up here in the chat. So if you would like me to read them, or if someone else would like to read them, we can start with some of these questions. Thank you so much.
Thank you so much, Jeff. That was very bio-inspired and inspiring, and I'll help you out with the questions. So please, everybody, just write them either in the Q&A section, or I've seen some in the chat as well. I will check both. So let's start with one from Stephanie. She says, of course, very exciting presentation and very inspirational. And with all the things you are handling, from research, grant writing, translation, lifelong mentoring of your trainees, how do you manage all of these? Do you just not sleep? But joke aside, how do you prioritize and delegate to make all this work?
Yeah, I think, this has changed, I think, over time. So early on, I wrote a lot of grants, like probably my first 2.5 years, I may have submitted like 100 grants or so. Most were rejected. And then I realized, it's more about kind of engaging the process of trying to learn how to write effective grants. And I started to collaborate with people who were successful at writing grants. And so that was kind of the early phases, was just kind of figuring out how would I get my lab funded. And so that's where a lot of my initial focus was the first couple of years. And then as, I kind of went on, it was how do I bring people to the lab who just have enormous potential, who with this environment of, empowerment could then start to spread their wings. And then some of the people in the lab would then be able to start, helping to write grants. And then as people started to develop further and further, they were able to, start to lead projects as well. So, we have had a number of projects from industry, and I like to have, people in the lab lead the projects as much as possible. So I'm in the background, sort of working with them, going through slide decks before we have meetings, but they're the ones in the meetings who are actually leading the the updates and the presentations. And so that's one of the ways that I've tried to, so I think, delegation is correct, but I kind of look at it more as, it's interesting because the word delegation misses a few key points, because it's not necessarily just delegating, because there's a lot of skill building that's happening when I kind of look at it more as like empowerment. So like if someone's going to lead, if all of a sudden there's a PhD student or postdoc in the lab who's leading a collaboration, they're really going to put in the time themselves to prepare, and they'll have a lot of questions, and there'll be a lot of learning in that preparation. And so I feel like that's one of the ways that I've been able to do lots of things is over time figure out ways to bring great people to the lab who have potential, find ways to unleash that potential, find ways for them to lead. And then that frees up some of my time to be more like guiding. And I think one of the ways I've also done that is through creating a lateral environment in the lab. So I really like to, I feel like my role is to help people maximize rigor and impact. But I'm also learning from the people in the lab. So trying to keep things as laterally as possible. Also, people are more likely to then own their projects and really, move forward. So I think, like sharing of credit, and whenever we have a press release, I try to get, whoever was involved in the lab to give quotes in the press release. So it's just, showing people appreciation and sharing of credit. And I feel all of these things kind of work synergistically together.
Yeah, that's wonderful. And we have maybe one last question from Amy. And Amy asked her question very early on, so I believe you already answered it, but I will read it and I'll complete with a little question of my own. So she asked, many basic scientists do not think about the translational commercialization potential of their work. Do you recommend working with your business development unit early on at the start of a project design, for example, to assist in formulating a translational strategy? And I think you already answered, but if you want to complement your answer, and I would ask, how much do you think the environment is? You're in Boston in a real hotspot. Do you think a driven individual like you could have achieved what you achieved anywhere else, or if the hotspot of Boston really contributed?
Yeah, so I would say that well, one of the first things that I did was when I started my faculty position is I went to the innovation office, the tech transfer office, and met the people there, because I feel like it was just really important to develop the right relationships. I wanted to learn how to do patent searches and just the process of how things work. I would say that I didn't talk about all of the failures and I talked about some of them, but at one point, actually, the tech transfer office said to me, you're not commercializing anything, and you filed a lot of patents and taken up a lot of our resources. We're not going to file any more of your patents. So that did happen at one point. The head of the office did say that to me. So, there's definitely been resistance, but I would say, that I also look at, a PhD degree it's a training degree. And so, you don't necessarily want to have too much of a commercial goal as an endpoint, but I think, having said that, developing skills in translation and sort of understanding what it takes to move things forward can be really beneficial and help you set you up for, future things that you're going to do. So I think any opportunity you have to, even check out like what's going on at the business school, or go, kind of see some of the talks that are going on, these entrepreneurial talks or these startup stories, and developing relationships with people who have done these things, I think could be really helpful as you move forward. And again, I want to emphasize basic science is really important, and it's a first step to, you can't translate anything before the basic science is in place. But I think if people are interested in translation, I mean, another thing you can do is there's like business plan competitions, and you may say like, okay, well, I have no skills to do it. But I think that's almost the point, is that you want to just jump in. And when you jump into things that you almost feel unqualified for, I think it creates the greatest learning opportunities. And then I think to answer your question about Boston, I mean, my sense is that every place has their kind of core strengths. Boston's a place where there's a lot of core strengths, but I think every place has, has a certain number of them. And so, my sense is really just to kind of hunt down, who's doing what in your kind of backyard, because there's usually examples everywhere of entrepreneurs and translation happening, and try to develop the networks, with those people. And I would say, as great, maybe kind of looking at Boston, as great as it is, I mean, there's also so many inefficiencies in the system, and it's still that, translation is really, really hard to do. And, I tell you, every single thing that I've tried to translate, at one point I thought was going to fail, it was going to blow up, we were going to run out of funding. Like every single project, it's always like that. Like, oh, I found this competing technology, and what are we going to do? And like, there's all these, things that we almost have like these meltdowns. But I think it's about kind of, appreciating time, and how time, there's a certain cadence to things, and sort of recognizing the learnings and the insights, and sort of just keep building incrementally every day.
Well, thank you so much for sharing your work and your passion with us. And we have a round of virtual applause.
Thank you.
And, I think we'll go straight to Nika to go on with the program. Thank you again, Jeff.